All right, I just want to thank you, everybody, for being here tonight. My name is Trey Kennedy. I'm an AmeriCorps member serving with Conservation Nebraska's Common Ground program. Conservation Nebraska's mission is to protect our state's natural legacy, and we do this by educating the public, supporting communities, and increasing community engagement, um, which we accomplish through these locally led education and community efforts. Before I introduce tonight's speaker, just a few things that we need to go over. Zoom audience. Your cameras are going to be turned off for the duration of the presentation. If you have any questions, please type that in the Q&A chat, and then we will also have a brief time to go over this um, at the end of the presentation as well. Dr. Kazi, if you'd like to have questions answered throughout, that is completely up to you, or we can save questions to the end. And then we will also ask to do a brief survey at the end. We do this to kind of gauge just how our presentations are going. And tonight's presenter is Dr. Kazi. He is a postdoctoral researcher at University of Nebraska Lincoln. He specializes in the release of nano and microplastics from plastic packaging under real-life conditions. His research employs experimental studies, systematic reviews, and meta-analysis to focus on the implications of nano and microplastics for ingestion of human health. He also holds a PhD in civil engineering, water resources from the University of Nebraska Lincoln, a master's in civil engineering that is in environmental engineering from Florida Atlantic University, and a bachelor's in soil water environment from the University of Dhaka. His work has contributed to the understanding of human exposure to microplastics and nanoplastics. Dr. Kazi, if I am missing anything else, please feel free to insert that now, but the floor is yours.
Thank you so much, everyone, for joining us today. And I'm really excited to present our work on nano and microplastics in food and what does that mean for human health. And thanks so much, Trey, for the nice introduction. As Trey was saying, I'm Kazi Albab Hussain. I'm from Bangladesh, and I have been in Nebraska for more than 6 years. And during my time in Nebraska, I have been working with nano and microplastics in food, and I would like to share some of that with you all tonight. So this is our very diverse group of experts from University of Nebraska system to tackle this challenge of micro and nanoplastics. We have experts from civil and environmental engineering department, from electrical engineering, from material engineering, from sociology, from food science.
Kazi, sorry to interrupt. I don't think your screen is shared.
The screen is not shared.
I do not see it. If anyone else can see it, please type in the chat, but I cannot see it. Patrick, can you see it?
Yeah, yes, I can see it.
Okay, then I guess my computer is missing, but that's okay if everyone else can see it. Yep, there we go. Now I can see it. Okay, all good.
Okay. Yeah, and we have also experts from public relations, from medical college, and youth and family studies. So we have a very diverse groups to tackle this problem holistically. Sorry, I have a hard time changing the screen. Yeah, sorry for that. And in this presentation, first, we are going to talk about what is nanoplastics and microplastics, and how they can enter in our bodies, and then nanoplastics and microplastics presence in our food, and then what is the health effect that can have in human, and some of those strategies that we can use to minimize our exposure to nano and microplastics. And at the end, I'll be happy to take questions from the audience. Now, our first topic is what is nanoplastics and microplastics. So nanoplastics, as the name suggests, smaller nano, and then the plastics means like the plastic particles. So, and if we define them in terms of the size, the nanoplastics are 1 nanometer to 1,000 nanometer. Now to put that in perspective, so the thickness of human hair is very, very thin, right? And well, the largest nanoplastic is 1,000 times smaller than the human hair thickness, and the smallest one is 100,000 times smaller than the thickness of a human hair. And for the microplastics, so they are 1 micrometer to 5,000 micrometer, or 1 micrometer to 5 millimeter. And the largest of the microplastics can be the size of a rice grain or the sesame seed. And the smallest one is the size of a bacterium or a fine toner powder. So the size of them are really important to understand, because this plays the major role how they're gonna behave inside our human body, or even in the environment. And as they become smaller, the problem for us become bigger. And there are primarily two types of microplastics. One, so primary microplastics, they are engineered. They are manufactured as micro or nanoplastics. And then we have the secondary one. And the primary one is actually, it can be in our like toothpaste, personal care product, cleaning agents, or cosmetics. And however, many countries have banned the production of primary microplastics, including US. And then the major problem is the one that ones are secondary ones. They are formed from the plastics we use every day, such as we used is the single-use water bottle. We throw it in the environment. Then what happens? The sun rays come, and then it exposed to different conditions. It breaks down. It started to break down into smaller, tiny pieces. And then those are called the secondary micro or nanoplastics. The problem with it is like even some of the plastics degrading is generating the secondary microplastics. But when we look into those packaging or the plastic products, it would still look like as a new, but it's degrading, it's generating micro nanoplastics. And this in this presentation, we are primarily going to talk about the secondary micro nanoplastics. And then how the micro nanoplastics look like. So they can come in different size and different shapes. They can be fragments like amorphous shapes. They can be like foams, or like threads, or pellets. Those are like the spherical shape. They are primarily used for the production of plastics. Then it can be the fiber. Lots of those fibers are generated from the laundry washing machine, and even it can be generated from our day-to-day use of our clothes. And then there can be like films, smaller films. Now in these slides, we can see them in very bigger size, so that we can understand them very clearly, that they can come in different shapes. But in reality, they are way, way smaller than this. Like maybe 99.99% of the micro nanoplastics we deal with are smaller than those particles. And it is so important to understand they comes in diverse shape, because the shape plays a crucial role how they're going to behave in the environment, or how they're going to behave inside human body. And now we know like what are microplastics. It can be smaller particles. It can be up to the 5 millimeter. Now we need to know like how they can enter into our bodies. So like think about your daily life. So you are getting out of the home, or even inside your home, you are breathing air. Then you are eating food, you are touching surfaces. And what you might not notice, that through all those actions, there are some micro nanoplastics are making their way into your body. Such as think about our first route of exposure, inhalation. So you are breathing some air. Since we already know that the micro and nanoplastics can be very tiny particles, so they can be suspended in the air, and when we breathe the air, they can go inside our body. And then comes to our second route of exposure is absorption through the skin. So since they are very tiny particles, they can get absorbed into our skin and make the way into inside our body. And then comes to our final route, which is the oral ingestion. That's the most biggest route, and our discussion of this presentation is to primarily be focusing on the oral ingestion. So when we take food, it actually, or drink water, take beverages, it can go inside our body. So now I will talk about some of the common sources of commonly detected micro and nanoplastics in some of our food, and how they go into the food chain. Such as we use some plastics. What happens next? We throw them, and after throwing them, they can break into small pieces. Like, yeah, say with some of the plastics are dumped into the ocean, then what, or they're dumped into the agricultural land or normal land. Then the sun rays comes, rain comes, and then freezing, and then there are the loss of heat exposure. All those contributing to the degradation of those plastics. And from the degradation of those plastic, what happens is smaller plastic particles get released, and those smaller particles can be washed if it is generated in the land, or if it's already in the water, then it have generated in the water. And then what happens is the organisms living in the water, such as fish, they can actually like confuse that as a food and then take it. And then when we eat the fish, we can actually eat them from the food. This is how it transfers from one trophic level to the another trophic level. So now this one is a very simple diagram explaining that how it can come to we can absorb through the food chain. And then this one adds another layer. We can see like the plastic packaging generating, we are producing them, and then we are using them to package many foods, like feeding bottle we can see, or like our packaged food. All of those can be the source of our exposure to micro and nanoplastics. And I'll explain that in detail how those can play a role in our exposure to micro and nanoplastics. So now there are some commonly detected micro and nanoplastics in some of our common foods, such as micro and nanoplastics were detected in common vegetables and fruits, and such as lettuce, carrots, onion, cucumber. All of those foods, scientists have found presence of micro and nanoplastics in those vegetables and fruits. And now they have probably deposited from the environment or introduced when we have packaged them, or maybe the root got it from even from the soil. So all those the ways the vegetables and fruits can accumulate those micro and nanoplastics. Now comes to the seafood. This is a very common source of micro and nanoplastics for human exposure. So in our ocean, we have lots of plastics that get degraded and it's generate micro and nanoplastics. And fish and oysters or shrimp can mistake them as a food and eat them, and this way they can make into our plate. And then comes to the infant food, and microplastics was detected in infant formula as well. So, and that can come from the packaging, or it had probably introduced when that was produced or processed. And in our drink, tap water, bottled water, drinks, tea, all of those beverages have been detected microplastics in all of those beverages. And such as for the tap water. So tap water were like comes from some sort of source, either surface water or groundwater, and then it's purified and sent to our water distribution system. Now the problem is like when the water purification happens, the purification systems are not well equipped enough to remove those micro and nanoplastics because of their tiny size. And also for the bottled water, they are sourced from groundwater or some sort of spring or somewhere. If it's from groundwater, it can have micro nanoplastics already in the source. And then the purification system, such as reverse osmosis or other water purification system, is not equipped enough to remove them from our bottled water as well. And also the bottled water comes in the single-use PET bottles. So that can be another source of the micro nanoplastics to our bottled water. And then all type of packaged products come in plastic packaging. And then the packaging itself made of plastics going through lots of pressure and movement and everything can introduce, and then storage and everything can introduce lots of micro and nanoplastics in our food. Now the question is, what is the number of the micro and nanoplastics that have been detected in all those kind of food sources? And the answer is it varies a lot depending on many things. First variables comes from the study that have how smaller of the particles they have detected, whether they have just detected microplastics or they have included both micro and nanoplastics. And if they have detected microplastics, what was the lowest size they were able to detect? Because of their smaller size, scientists often fail to detect the smaller ones. That's why they just report the bigger ones, and that's why often the concentrations comes in lower number versus the if some other studies detected all the range of the particles, like starting from 1 nanometer to 5 millimeter, then it would be way higher than the one which has just detected the microplastics or the bigger microplastics. Now, so now I'm going to talk about some of the research what we have done at UNL. And this one talks about how plastic food containers can introduce micro and nanoplastics in our food. And this study was published in the Environmental Science and Technology, the best journal of our field. And since the publication of the study, like the news channels from all over the world covered this study because it's in such an important topic, and it have actually addressed some of the very common route of micro and nanoplastics exposure for our everyday life. So we have selected 3 food containers. So 2 made of polypropylene and 1 made of polyethylene. So we have tried to address few questions. So first, we wanted to see whether those containers and reusable pouch releases any micro and nanoplastics into the baby food. And if the release, what's the quantity of the release? And then we wanted to see whether those released particles have any negative human health effect. So this is a video. This is the first qualitative evidence that the micro and nanoplastics release from our containers. In the first video, those we see very few white dots. But in the second video, we can see lots of white dots. Those white dots are actually particles. In the first video, we see very few white dots because this water did not came in contact with the plastic. But the second water, the video was taken from the second water, that one came in contact with the plastics. And we have seen, like, after coming in contact with the plastics, it has way more particle than the normal water. In our normal water, even we had some particles, but when it came in contact with the plastic, we have seen the concentration increased many more times. And this video was taken using using an instrument called nanoparticle tracking analysis. This one uses some light scattering techniques. And then we have exposed those containers in different scenarios. First scenarios, we have wanted to see like what would be the release if we store food using those containers into the refrigeration. And then in second scenarios, we have tested what would be the release if we stored them in room temperature condition. And in third condition, we wanted to see what would happen if we expose them in high temperature. And then fourth condition, we tested them for the release in microwave heating. So and this one is actually, we have followed the United States FDA guidelines to replicate the release for 6 to 12 months period of time. And what we have found, the release can be thousands to millions of microplastics just from 1 square centimeter of the area of the container. So, and this one's just talking about the particles that are bigger than 1 micrometer. And it covers the whole range of microplastics. And we have also found comparing all those conditions, we have found microwave heating releases the highest number of microplastics compared to the refrigeration room temperature or high temperature storage. And we have also seen one thing, that the release varies when we change from one type of food to the another type of food, such as in here in the red one represents the aqueous type of food. Then in the blue one, it represents the acidic type of food. And we have seen in the room temperature condition, when we're changing the food type, the release is changing drastically. And also it's true for the microwave heating. It's also behaving differently when we are changing the food type. Yeah, sorry, I'm having an issue to change the slide again. So, and then we have tested for the nanoplastics release. And what we have found is, and the release can be from millions to billions. So the release increases by many-fold when we count the smaller particles. And again, we have found the same thing. The microwave heating releases the highest number of particles. And then we have actually analyzed what's the size of the particles. And we have seen like when we consider the smaller portions, their number is very high, such as for the nanoplastics, we can see like most of the particles are within 400 nanometer. So 10 nanometer to 400 nanometer, most of the particles. And that means they are very tiny. So that means they can make their way into our body. Even if they are inside our body, they can make way inside our body very easily. And for the microplastics, most of the particles are within 3 micrometer. And in this video, I can actually show you like how we have confirmed this, the particles we are counting, they are coming from our bottles. So in the first video, you can see we have an empty bottle. This is an schematic filled with water. Then we have microwaved it for 3 minutes. And then we can see the waters with lots of particles. So now in this video, you can see, in this picture, you can see a clean gold coated filter. And then we have filtered 300 milliliter of water. And in the picture, you can see we have actually accumulated lots of particles. Those are small particles. They have accumulated, they have got released from the water, and we have actually accumulated them in the filter surface. And then we took this filter to analyze using the Raman. And then from this signal, we can confirm that the peak matches the properties of polypropylene polymer. So that confirms that the particles were indeed coming from the polypropylene made container. And then we have went for to estimate their exposure. Now, when it comes to the exposure, we have used the release data from our tested containers, and we have found that the release can be when we count it in terms of numbers, can be billions to trillions of nanoplastics depending on the what is the what product we are using. Such as if we use those containers for water versus for the dairy products, the ingestion increases at least 20-fold. And we have seen just using those containers can result in the ingestion of billions to trillions of micro and nanoplastics. And this is a very interesting slide. So we have talked about like there are lots of micro and nanoplastics getting released from those containers. So we have used some techniques to extract those particles. So we have actually freeze-dried and lyophilized them to extract those particles from the water. And then we have used in the lab, we have grown some cells. And in the first picture, we can see that human embryonic cells are growing nicely, happily, they are forming communities. And then in the formed communities, we have actually introduced food. But in the food, in first one, we have just introduced the food for the cell, the regular food. And in the second one, we have added food and the micro and nanoplastics we have extracted. So in this picture, we have actually added 250 microgram per milliliter of nanomicroplastics. And you can see in the second picture, most of the cells, they got dead. And however, in the control cells, they were forming nice, happy community. But when they have the micro and nanoplastics with them, so most of the cells were dead due to the presence of the micro and nanoplastics. And some of the take-home messages from the findings of our study, that the food containers can play a significant role in our exposure to nano and microplastics. And the result indicated that microwave heating can cause the highest release, such as we have tested like room temperature storage, refrigeration storage, microwave heating, and we have found microwave heating causes the highest release. And then we see like what type of food is coming in contact, such as aqueous type of food or the acidic type of food. That also plays a crucial role in determining how toxic, how much of the micro and nanoplastic can get released. And then we have extracted those particles and we have conducted the cell viability study, or we wanted to see whether the cell can survive when there are micro and nanoplastics present. And we have found that they can cause the death to the human embryonic kidney cells. Now, from some other studies in the literature, we can see what other packaging can be a problem for our day-to-day life. Now we can see like some of the common plastics that are used in our everyday life, such as the single-use water bottles. They are made of PET or PETE. Then the biggers like the milk bottles or the bigger water bottles, they are made of LDPE or HDPE. And some of the bags we see every day, they are made of LDPE. And then the food containers we use, they're mostly made of PP, and then those are made of polystyrene. Now, why this one is important, that what type of plastic they are made of. So from our experience, we have seen that it depends how much of the micro and nanoplastic would be released. It can be related to what type of plastic they are, what type of polymer they are, such as whether they are made of PP or PE. I mean polypropylene or polyethylene. Now the question might be like, which plastic releases less, which plastic releases more? So these are the some questions we are trying to answer in our lab. We are doing lots of studies to find that. But based on the studies from all over the world, we actually try to answer that, but it's actually no definite answer is not available at this moment. But one thing we can confirm that the release depends on the polymer types, and it can also depend like which type of some polymer may be good for one type of application, then the other polymer may be good for the another type of application. So it's really important like which type of polymer being used for which type of applications. So one of the aim in our lab is to trying to find the safer type of polymer for the different type of packaging. And now from the literature data, we have seen the lots of beverages such as bottled water. Bottled water can be a common source of micro and nanoplastics. And it can, such as the bottled water is coming in a plastic packaging. The packaging can be a source, or it can come from even the source water. And then the tea bag. So studies have found the steeping process of the tea bag in hot water can release of millions to billions of micro and nanoplastics. And pouring the hot beverage in paper cup or plastic cup can lead to the release of micro and nanoplastics from those. The paper cup has a lining which is made of plastic. So, and this lining can release micro and nanoplastics. And in our kitchenware, such as we have cutting board, the striking of the knife into the cutting board can release lots of like millions of micro and nanoplastics. And then the blending process can release, it's very forceful process, and that can also release lots of micro and nanoplastics. And the kettle made of plastics goes through lots of heat and can generate lots of micro and nanoplastics. And then comes to the convenience food, such as like the takeout food container. The pouring, like the storing or carrying the hot food into the takeout food container can leads to the release of micro nanoplastics from the takeout food container. And same thing true for the instant food container. So when we instant food, it's often involves some sort of preparation into most of the cases into the oven or into the microwave. So such as instant noodles, that can be a very big source of micro and nanoplastics ingestion for human. And then the baby products, such as the feeding bottle or the tip, they can be a significant source of micro and nanoplastics. Studies have found millions of micro and nanoplastic can get released from feeding bottle when they pour the hot water into it and the formula preparation itself. And in our study, we have found food container and the pouch used for the baby food can be an important source of micro and nanoplastics exposure for babies. And the other type of packaging, such as rice packaging, fish packaging, or the chicken packaging, they have the plastic film and the plastic tray. That can be an important source of exposure. Now, again, if we talk about the concentration, it depends, it varies a lot depending on the packaging type, what conditions they are getting exposed to, such as someone's exposing it to very high temperature versus someone just exposing it to room temperature. That will determine a lot how much of micro and nanoplastic they will get exposed to, or that will get released from that particular type of packaging. And in the next slide, we're gonna see like what are the how much of the micro and nanoplastics we can get exposed from different sources. So, and we can see like the yearly exposure of microplastics from outdoor air can be a few thousand, and indoor air also a few thousand. And we can see like from the other sources, it is few thousand. So now I have been talking about like millions and billions, but in this study, they have shown like just thousands. So the difference between what they have covered here and what I was talking about is you probably already know they are just including the microplastics. So, and in the next slide, we can actually see like when they include, if they have include nanoplastic, what the number would be. And also we can see like they did not include the packaging as a source. So if they have include packaging, the different type of packaging, so that number would be really, really different. So the answer for this question is like how much of micro and nanoplastics we get exposed to. We do not know that yet. And we do not know that yet because of many reasons. I'll cover that in a later slide. So in this slide, we have seen this number is underestimated because of the few reasons, like we have analyzed the study of the takeout food container. And we have found like when the studies are using lower detection limit, means when they are reporting particles of smaller size versus when they are just reporting the bigger sizes. This number changes linearly. So studies with smaller particles always report higher concentration than the studies with just the big particles. And that means when if we have included the nanoplastics, the number would have been changed drastically. Now we know that we can get exposed to those micro and nanoplastics. And what does that mean for our human health? A common assumption can be like we are getting exposed to those micro and nanoplastics. So lots of them just can get out of our body. But that's actually true if they were just bigger size. The bigger ones probably getting just excreted from our body. But however, we consider the smaller ones. Since they are very small, they can actually cross the barriers and get accumulated in different organs. And in these slides, we can see actually like the researchers have detected the presence of micro and nanoplastics in different human organs, starting from the brain, lung, stomach, breast milk, liver, kidneys, and even in our skin. And that numbers actually varies a lot, and we do not know what the exact number of accumulation in different parts of human body because we do not have the techniques to detect the smaller ones in complex matrices. And now if we get exposed to those particles, what are the common problems we can have? First of all, like we can have inflammation. So when our body get exposed to some foreign objects, such as some virus, they start to fight against it. Now, the same thing happens when we get exposed to micro or nanoplastics. So our body treats as a foreign object, and they try to get rid of it. And it's not easy to break them since they are like polymer. Polymer is not easy to break. So during this process, it actually causes inflammation, and it can be very damaging for our tissues. Then when the micro and nanoplastics get introduced in our body, that introduction can generate lots of free radicals. And during the process of trying to break it or trying to get rid of it. And this generation of free radicals can actually cause oxidative stress. And that oxidative stress can actually damage cell and damage the DNA. Now the accumulation of micro and nanoplastics in different human organs can impair their function. So it can, such as in previous slides, I have talked about their accumulation in brain and their accumulation in lung, and that can impact the normal function of that organ. And then that can also affect the blood circulation. In a recent study, the researchers have found that the people with stroke have a higher percentage of accumulation in their blood circulation system, micro and nanoplastic accumulation in their blood circulation system compared to the healthy human. And also exposure to micro and nanoplastics during the pregnancy can impact the fetal development. Now some of the questions about the about the health impact of micro and nanoplastics. Do we know a lot? The answer is no. We don't know a lot yet. It's very tiny amount of the knowledge we have about the impact of micro and nanoplastics on human health. And the reason behind it, we have very limited data on the release of micro and nanoplastics from food packaging. So, and we also have very insufficient information about the occurrence of nanoplastics in food. Like I was saying throughout the presentations, their size, the nanoparticles, they are being so tiny, make it so hard to count them or to detect them in different complex matrices. And also, whatever we know about the health effects is actually from the model particles based on some engineered particles. But when we get exposed to amorphous shape of particles and in a mixture of different size of particles. And so it becomes really complex when we get exposed to those particles in real life versus than some ideal particles. So the mixture of particles will have way different effect than an ideal particle. So those are some limitations actually uh, hindering our way to get a vast knowledge about the human health effect. And finally, I would like to share some of the strategies based on the knowledge we have gathered that will help us to minimize our exposure to micro and nanoplastics. First of all, we can avoid we can choose fresh food. So we have already learned from our presentation that the packaging can be a source of micro and nanoplastics in our food. So simply avoiding them can be a way to avoid the exposure to some of the micro and nanoplastics. And drink tap water when safe. So we have found from the literature that the tap water has lower number of micro and nanoplastics compared to the bottled water due to the many reasons. And so drinking tap water can decrease your exposure to micro and nanoplastics, but you have to make sure the drinking water is safe from other type of like contaminants. And using glass or stainless steel or ceramic containers. So we have seen from our research that the plastic containers can be a big source of micro and nanoplastics. So using glass or stainless steel can avoid that source. And also we have consistently seen from all the literature that when we are using plastic for high temperature, it releases a lots of micro and nanoplastics. So for when it comes to using for the high temperature, avoiding plastic can save us from exposure to micro and nanoplastics. And we have also seen when you store the food for long term into some plastic container, with the time increases, the release of micro and nanoplastics from the container increases. So for the longer storage, avoiding plastic can save us some of the common micro and nanoplastics exposure. And I would like to thank those funding agencies that have supported our research at University of Nebraska Lincoln. And I would like to thank you all. And I'm ready to take any questions.
All right, wonderful. Thank you, Kazi. That was a great presentation. Definitely some things, some food for thought. We did have a couple questions for you. One of them was earlier. And is tea bag made of paper or is it made of plastic?
It is most of the tea bags. It looks like made of paper, but it's actually made of blend of polymers.
Gotcha. And
Yeah.
then we did have another one. Do we have any way of effectively filtering out the larger microplastics?
If it's very large, definitely yes. And then, but if it's the smaller one, say like the nanoplastics, It's actually, we do not have the filter of that capacity that can actually eliminate those nanoplastics. But for the larger ones, yes.
Then another one we had in the chat was any concerns with silicone bakeware?
Silicon is also a plastic, and we actually like some of the studies actually found the release from different type of silicon materials, but we are still in the process of seeing actually like whether silicon do any better than the other type of polymers. But this is a very good question.
And then does cold temperatures also increase the plastics that get into food and water? Or I guess, is there any temperature difference that affects it?
This is also a really good question actually, and the answer is like we have seen the increase of the temperature always increases the release. When it comes to the like using the room temperature is probably the best one, but we're actually unsure about the like if we what would happen if we store them in the like in the freezer. So this type of things are still kind of in the process of knowing actually.
Then I actually had a question. Is there any research being done with plastic eating bacteria for
Oh yeah,
microplastics?
this is a good question. We have seen actually like lots of research when it comes to the degradation, but actual expertise is not in that part of degradation part, like, but it's actually often comes to the like when we read different literature, we've seen like there active research going on. Probably even at UNL, one professor is doing some study to see whether some of the like microbes can degrade the micro and degrade the plastics effectively.
Wonderful. And then it looks like we also have a couple more questions. Are there any certain industries that contribute microplastic pollution on a larger scale?
Yeah, so actually, like we can tell like this, and now if we say like we are using all types of plastic packaging, and then it's everywhere in our life, right? So then I think the plastic packaging is a big source of our exposure to micro and nanoplastics. But certain industry, I'm not sure.
And then this might be a follow-up to the silicone question, but is Teflon covered bakeware a problem?
I'm not sure about that. I have not noticed any research regarding that, like the Teflon cupboard, but this is definitely a good question, and I'll look into it actually to see whether Teflon covered bakeware is any safer.
Wonderful. And then is research for microplastics under any danger of losing funding as a green industry? Quote unquote green.
Yeah, so this is a very interesting topic, but I don't know. Like we have seen a shortage in terms of like research for every field comprehensive, but I have not noticed any news regarding that. Like they're gonna cut funding for the micro nanoplastics research specifically.
Understandable. Looks like the questions have stopped coming in. Um, yes, we will also send out a link to this recording. It will be up probably in the next week or so. We just need to get it finalized. But it should be up relatively soon. Um, is there any other questions for Kazi?
There was a follow-up question in the webinar chat relating to food in containers.
Um, what if a plastic does not touch food or water? For instance, if the is glass but the lid is plastic.
And this is a good way to go about it actually. So so I think is actually since it's glass, then we are definitely sure that the the plastic particles are not coming from glass. But if it's like, if we put it into the microwave, then we do not know like the lead is leaching any micro or nanoplastics. Maybe yes. But generally, if it does not come in contact, I would think it's a better option. But this is a really good question.
Is there any last questions for? Oh, and then sorry, we did just launch the feedback survey. If we can get quick 30 seconds for everybody to quickly fill that out, that would greatly help us. It shouldn't take too long. But Dr. Kazi, is there any just final thoughts, comments that you'd like to make closing out that you think would be important to share?
I think we covered a lot so far, and I really liked the questions everyone had.
Good, good.
And I appreciate what you guys are doing.
Wonderful. All right. Well, Dr. Kazi, greatly appreciate you coming out and taking your time to do this. You shed light on an issue that is probably not being talked about enough. But I definitely learned a thing or two, and hopefully everyone else did too. But thank you for coming and everybody that attended this evening session. So we will close out.
Thank you, everyone.